Some of them are just, you know, you gotta think your way through the question. And you know how to write a genus and species name. You know what colonial morphology is as opposed to cellular morphology, right? Colonial, I see colonies on my feature plate. Cellular morphology, I see the cells in my microscope. And okay, let's see. A couple more questions. Don't forget your growth curve, right? Your bacterial growth curve. Bacterial population growth on a limited medium. It's not going to be there forever. It's going to run out. So you have your lag phase, log phase, stationary phase, death or decline phase. They're going to eventually use up the agar, or use up the broth, or use up that slice of pumpkin pie that's been in the refrigerator since last Thanksgiving. Can you believe how close we are to next Thanksgiving? Oh my God. I went to Michael's and they already got the Halloween stuff out. And I would buy some of it, except I already have 20 boxes of Halloween decorations. It's ridiculous.
My favorite holiday.
Yeah, me too. My favorite one, because candy rules. It's all about the candy. And the orange and black, which is tiger color. So, alrighty. Okay, so you have to know the phases of your growth curve, what's happening in each phase, what's the healthiest time for the bacteria, when would they maybe make inclusions, when would they be acclimating, that kind of stuff. Running an active metabolism. Alrighty. What happens in transverse binary fission? Parent cell elongates, copies its nucleoid. Nucleoids go to opposite ends of the cell as it builds that transverse septum or transverse cross-wall plate, whatever you want to call it. Then the two cells separate into genetically identical daughter cells. But what does not happen during transverse binary fission? No genetic recombination. The daughter cells are clones. If I do get a little bit of change, a little bit of genetic recombination, what caused it? Mutation. Right. So your gene transfer mechanisms, your horizontal gene transfer mechanisms, conjugation, transformation, transduction, that's going to happen at a separate time. It's separate from binary fission. Okay, what's MRSA? 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 Good. What's VREF? What's MDR-TB? Good. What's CRKP? That's CRE. KP would be Klebsiella pneumoniae, which is one of the Enterobacteriaceae. But CREs are like carbamazepine-resistant Enterobacter species, or it could just be Enterobacteriaceae. Everybody uses it a little different. All right, we classified our bacteria according to the old version of Burgess' manual, which was made in the the 1940s, I think it was. And we classified our bacteria into 4 divisions. Division 1 is the gracilicutes, the gram-negative and thin wall. Division 2 was the firmicutes, gram-positive, thick wall, including acid fast. Division 3, tenericutes, which have no cell wall, those are your mycoplasmas. And then we had Division 4, which is not medically important. Those are bacteria that are extreme types. And that one is called Mendoza cuties. Is that right? Yeah. So, your textbook, however, uses a more modern classification for bacteria, because instead of classifying them based on structure and function and metabolic characteristics like we've done in lab all semester, they classify bacteria by looking at the sequence of genes in the ribosomal RNA. They are looking at RNA letters. And if they have a high number of G and C letters or a low number of G and C letters, they put them in different groups. And if they match, they put them in the same group. If there's not much of a match, they put them in a different group. So our method of classifying bacteria was based on their phenotype. The new method that your textbook will use is based on genotype of ribosomal RNA. Our RNA is what they're looking at, not DNA, which is pretty strange, but that's what they do. And what did we call the groups in the new classification scheme? What are your gram-negative bacteria called in the new classification scheme? What are your bacteria called in division 2 in the new stream? What? Oh, I don't think I have it with me anymore. Oh, yes, I do. Your handouts package, please. You could go back to the PowerPoints or back to the textbook and look, and it's all there. But you can also do this. Back of your handouts package, remember the moldy bread? Here's your bacteria. Proteobacteria. What do we got there? Campylobacter, Helicobacter, Pseudomonas, Acinetobacter, Burgdorferi, and those kind of guys, Escherichia coli, Haemophilus, Enterobacter, Klebsiella, Proteus serratia, Salmonella, Shigella, Yersinia pestis. Proteobacteria. The gram-negatives are proteobacteria. Firmicutes. Oh my God, isn't that what we called them? Yes. And now they're still calling them firmicutes. What do we got? Streptococcus, Staphylococcus, Enterococcus. Oddly enough, they throw mycoplasma in a different section. But so your firmicutes are gram-positive. Your gram-negatives are proteobacteria. And then they have a few other different groups.
What do you want us to know about this?
Yeah.
You need to know the new classification scheme, yes. I mentioned that before when you had test 2. These are viruses. It's the same information. It's just laid out in more of a tree fashion than your list. That's all. It's the same information. This is just more visual rather than a table. They set it up like trees. This is the classification scheme of your textbook. This is the current accepted classification scheme. It's all the same viruses, all the same bacteria, all the same parasites. Somewhere on there, I thought I put the website. It's called Biomed Central. I better make sure I have that on there somewhere. I print it up next time. It's from Biomed Central. I'll write myself a note about that. I didn't cite the reference. Hey, let's see what other questions we got. Again, don't forget about your Lancefield groups. Your Group A, Group B, Streptococcus. Which ones are which? Got some post-it notes down here. What's herpes simplex? Hope or if it's herpes simplex 1. Okay, above the waist, herpes simplex 1, below the waist, herpes simplex 2. And you know what a strain is? Strain and serovar are similar words, but not strand. Don't call it strand if it's a genetic variation of the species. And you know what coliforms are, right? That's E. coli and his friendly buddies. And when it comes to antibiotics, remember mode of action is how the antibiotic exerts its effect on the microorganisms. If the antibiotic targets the ribosomes, it's going to inhibit protein synthesis. That would be your tetracyclines. If it targets the peptidoglycan of the cell wall, those are your beta-lactam antibiotics. Penicillin, bacitracin, vancomycin. If it targets the plasma membrane, those are your polymyxins. Antifungals also work by targeting the plasma membrane of the fungi, and those are azoles, imidazoles. So it's got A-Z-O-L-E. Clotrimazole, can't think of another one right now, but they're azoles or imidazoles. And then there are some antibiotics that work by interfering with nucleic acid synthesis. Those are going to be particularly helpful with viruses. There's two ways that an antibiotic that interferes with nucleic acid synthesis can do it to a virus. Number one is base analogs. They're also called nucleotide analogs, nucleoside analogs, whatever. It's phony letters. The other way is interfere with viral enzymes needed for nucleic acid synthesis. Okay? And don't forget your escape pathogens. Who are your escape pathogens?
Superbugs.
Right, but can you name them? What's ESKAPE stand for? Yeah, you gotta go back and look them up. Right? I think that's it. Is that enough?
I thought it would be very general.
Yes, much more general.
Looks like it.
Much more general.
All right, good.
So you can take a breather and we'll start your lab practical.